We're almost there. There we go. Try that. Um, so these are the premises that have deer within the state of New York. The black numerals are the number of premises that are in an area. So for example, if you look here at between Rochester and Syracuse, this number 16 indicates that there are 16 deer herds in that area between Rochester and Syracuse. The single green inverted teardrop is just one herd up here. These, if you are working at all with captive cervids, I want to give you a quick tour of our Department of Agriculture and Markets page for captive cervids so you know it exists and some of what you can find here. So if you do an internet search for New York captive cervids, it will bring you to this page. This is the top of the page, but if you scroll down a little bit, you get to this health section, and it does have some resources for diseases that affect deer. You can see along the sidebar here that it does have additional information about identification, including this link here where deer producers can order official ID for their deer. We have our import and export regulations listed. There's information on programs and permits also here. And just below that, we get to this section here called Related Resources. And the related resources have some links that you might be interested in. Probably the most interesting one for you as veterinarians is this deer farming webinar by Dr. Douglas Wagner. So that is available if you're interested in listening to that. This recording will actually go up on that website as well, in that spot, so you can see it. It's also good to know if you're in the veterinary field and you're considering working with deer, they do get some diseases that are reportable in New York. So if you search the internet for New York agriculture and markets, reportable diseases, you'll find this page. And if you scroll down, you can find this reportable disease list, which you can click on it and download it. If you scroll down to the bottom of page 3, the cervid diseases that are reportable are listed here. And then if you are suspicious that you are dealing with a reportable disease, if you go back to the page you just came out of, down on the bottom of the page is this section, reported disease. If you are a veterinarian, you can give us a call. If you are a producer, you're going to reach out to your state field veterinarian and let them know. So backing away from some of the veterinary resources here, because we're going to ask Dr. Nichols to provide a much more interesting CWD presentation for us in a moment. Let's look very briefly at the captive cervid herd program in New York. Each herd in New York has a status for each of these 3 disease programs. I'm just going to quickly mention them as an overview. There's CWD, tuberculosis, and brucellosis. Again, this is going to be a very quick look at these. So each herd is going to have one of these 4 different tuberculosis statuses. They may be accredited to allow for the movement of deer. They may be qualified on their way to getting the full accredited status. Herds may also be tested, or they may be quarantined and unable to move live animals. Brucellosis in our state is a voluntary program. There are some herds that are certified or qualified to become certified, but many have no brucellosis state. This is helpful for folks who are exporting deer to other states. And then this is the 3rd disease program that each herd will have a status in. There was tuberculosis, brucellosis, and their CWD. So each cervid herd is going to fall into one of these categories. They are either a CWD susceptible species or they're non-susceptible. The CWD susceptible deer are going to either be a certified herd or a monitored herd. This is the beginning. This is a slide is continued on the next screen. These are some of the species that are defined by the USDA as being susceptible to CWD. So on this page in particular, they've got white-tailed deer, some elk and red deer, which are common species in our captive cervid herds in New York. This is a continued list. sika, some white-lipped Barasinga. We have some of these deer in our state also. I'm going to come back to reindeer and muntjac in a moment. If we get the presentation to move forward. There we go. Non-susceptible deer are going to not be biologically susceptible, we believe, to CWD at this time. We have a few of these species, but the most common by far in our state is fallow deer. Reindeer and muntjac, you can read through this slide. It has a little bit more information later at your leisure if you want. But the important thing for us in New York is we do not allow the import of CWD susceptible species. So at this time, because reindeer and muntjac are classified as non-susceptible, they are allowed to be imported into New York. But as that changes, because we expect that to happen within USDA, because they do seem to be biologically susceptible, their import will be banned in New York at that time. So of our 179 herds in New York, 77 of them are certified. So in a certified herd, any animal that dies or is harvested must be CWD tested. For the CWD monitored herds, they must submit 10% of their herd each year. So if they don't die on their own, we have to have them harvested by the producer. And then there are 30 CWD non-susceptible herds in our state. Some program history. In 2005 for CWD, 18 years ago now, some captive and wild deer in Oneida County were tested positive for CWD. There was a strong response from the New York State DEC and Ag and Markets, and no positive cases in New York have been seen since then. We are the only state in the US to have successfully eradicated CWD. If you do an internet search for New York Times 2005 chronic wasting disease, you will find this article that explains how a farmer donated some venison to a fire department banquet. Pardon me. And later that deer tested positive after more than 300 people ate some of the venison. So the Department of Health was heavily involved in following up with after this exposure. Still to date, no humans have become ill from this infected venison. And then, although we are still currently CWD free in the spring of 2021, there was a case of CWD within 5 miles of our border in Pennsylvania. So we feel it's important to educate people, including veterinarians, about CWD. So next we have Dr. Tracy Nichols, who is going to tell us more about CWD. And I think Tina is helping us organize this, and she put in the chat that if you have any questions, she put in the chat, if you have any questions, please put your questions in the chat and we will answer them at the end of this presentation. See if I can stop sharing. Actually, while I'm trying to figure out the buttons for that, I will give you an introduction to Tracy Nichols. Dr. Nichols is the Senior Staff Officer and Chronic Wasting Disease Subject Matter expert in the USDA APHIS Veterinary Services, and she is part of the Cervid Health Program team. Thank you, Dr. Nichols, for being here.
Thank you for asking me to come and talk about CWD. I actually very much like to talk about CWD. It's all I've done for about the past 17 years or so. Trying to get my presentation to go on presentation mode. There we go, much better. I'm going to turn my camera off to preserve some bandwidth. So I'm gonna disappear here. Are you able to see my introductory slide?
Yes, we can. Thank you.
Okay. So as Dr. Shane Holzer mentioned, I'm with the USDA Veterinary Services Cervid Health Program. We are the agency that is responsible for the rules and regulations regarding interstate movement of farmed cervids. My background, I'm a cell molecular biologist, and I've worked exclusively on CWD for about the last 17 years, initially as a research scientist. Then I took a regulatory position with veterinary services. See if it's going to play nicely with me. There we go. So when we think of diseases, we rightly think of bacteria and viruses, and we're all too familiar with viruses these days. But when we're talking about chronic wasting disease and its associated relatives, like BSE and Scrapie and even CJD in humans, we need to take what we know about those and crumple it up and throw it away. Because transmissible spongiform encephalopathies, or TSEs, that's the group of diseases called prion diseases. That includes Scrapie, Kuru in humans, that was in Papua New Guinea, bovine spongiform encephalopathy, or mad cow, CJD and variant CJD. Variant CJD was the human form of BSE. And chronic wasting disease are in this group of diseases that are not caused by a bacteria or a virus. They're very different. So in this cartoon, it's a schematic of the membrane of a cell, and attached in that membrane in a lipid raft are these things called prion proteins. And the prion protein is a normal endogenous protein. It's really highly conserved. There's even forms of it in things like yeast. However, the function is slightly different. So these are GPI anchored proteins, and they're expressed throughout the body, with the highest expression in the brain, lymphoid tissues, which includes the spleen, but you can also find it in other places if the animal is far enough along in disease, such as muscle, reproductive organs, and peripheral nerves. So this is a classic tale of when good proteins go bad. Excuse me. So the normal prion protein, as I mentioned, is endogenous and highly conserved evolutionarily, but it becomes a problem. And so what happens there to cause it from going to be a good normal functional protein to one that's disruptive and ultimately deadly? So in this cartoon, hopefully you can see my cursor. On the left-hand side, we have a diagram of what the normal conformation of the prion protein looks like. It has 3 alpha helices and 2 beta sheets. Okay, this is how it should look. However, it goes through a change of shape, and we don't, excuse me, we don't really know exactly what causes it to change shape. So if you remember back to biochemistry, proteins don't like to do that without some sort of catalyst or reason to do so. They like to remain stable. However, this changes from this normal shape to this aberrant shape. And so now there's only 2 alpha helices, and now there's 4 beta sheets. Beta sheets are generally a problem in proteins. They cause proteins to stack up. You can see beta sheets in things like A beta in Alzheimer's, tau protein in Parkinson's, and I think tau is somewhat also involved in Alzheimer's. They start to stack up, and that's a problem. And then once it changes shape, there's a core of this protein that's resistant to protease digestion, and that shouldn't happen. You should be able to put a protease on a protein and it chews it up. It doesn't chew up the core of the prion protein, and this is going to be a factor when we start talking about disinfection and degeneration of the prion protein, and why it's a problem. So chronic wasting disease is more common in deer than in elk. Not that elk don't get it, they absolutely do. There's just a number of things at play there that makes their prevalence rates not quite as high. It can also affect other more exotic species like muntjac, reindeer, cycadeer, and others. And I have a question mark there because we really don't know if some of these other species that you saw in Dr. Shane Holzer's presentation, we don't know about those because either we don't see many of them, or maybe they don't have contact with with infected animals. So that list could be expanding over time. It definitely has expanded since CWD was first discovered. Contrary to popular belief, CWD can affect animals of any age. This makes it different than BSE. So when we think about BSE, we think about older cattle, not the case with chronic wasting disease. We have seen CWD in animals under 12 months of age. It has a really long silent incubation period, which is one of the reasons like it's a rather shady disease. You can't pick it out easily. So when we think about CWD, here we have an elk that we had in a farm here in Colorado that was on study, and this guy has CWD. We had to take his antlers off because he was super aggressive. With elk, you see that. You don't really see that with deer. He's got droopy ears. He's a little sawhorse stanced. That's what we think of. However, most animals with CWD look like this buck down here because the incubation period is silent and they're asymptomatic up until the very end. So you have no idea who's got it and who doesn't by looking at them. And so you'll see this figure about incubation period, roughly 18 to 24 months. Yes and no. So what we've learned is that there are genetic factors that significantly alter this, and I'll talk about those a little bit later. So if you have your standard highly susceptible white-tailed deer, you have an incubation period probably about 18 to 24 months before you start seeing clinical signs. Once they have clinical signs, they usually die within a pretty short amount of time. So this kind of goes back to my previous point, which animal has CWD? We always think about this poor looking animal here, rough coat, head down. But most of them I've seen thousands and thousands of deer now, they look like this one on the left. They look great. And I tried to make notes during depopulation events to see if I could predict. I absolutely cannot. So some animals I thought looked a little straggly. No, they didn't have it. And other ones that looked really great and had great body condition did. So much for trying to, pick them out of a crowd. Terminal clinical signs include poor body condition, lowered head and ears, PUPD, ataxic. You'll see a sawhorse stance, drooling. Pneumonia, specifically aspiration pneumonia, is probably the most common reason that CWD animals die. They start having destruction of their motor neurons and they stop being able to swallow properly. So they get saliva down into their lungs. And you'll see sometimes a lack of fear where they should be afraid of you, but they're not. So how are they getting CWD? Well, there's two types of exposure. There's direct and indirect. And so this image on the left is a trail cam that we had here in Colorado between our farmed elk over here on the right, and she's getting a visit from two wild elk coming to say hello across the fence line. So we know that they will interact across the fence line. Whether that happens between deer and elk, I would think probably not. Elk will pick on deer if they get a chance. And then indirect is a nice picture of some soil and some grass, is environment. And I'm going to talk about each one of these. So we know that CWD can be transmitted directly nose to nose, but we also know that it can be transmitted in utero to babies. So while they don't have any symptoms, what we'll find is if we were to euthanize, let's say mama here had CWD, and we were to euthanize this baby 9 months or so along, we very well could find it. So definitely happens. There've been a number of papers that have kind of, I think, put the nail in that coffin once and for all that yes, it can be transmitted in utero. Because we used to just think it was horizontal transmission, but now we know it can be vertical. Then we go to environmental disease input, and there is a lot of components to this. So we have obviously carcass decomposition. So if an animal dies in the wild or a hunter chucks out a carcass that's positive into the environment, it degrades and goes into the surrounding soil and plants, and we'll talk about that in a minute. Scavengers like crows or coyotes, we've done studies on both of those to show that what goes in one end comes out the other. They don't necessarily seem to be affected by the disease itself, but they can translocate it in the feces. Here we have our droopy elk, and he's salivating, so we know there's CWD in the saliva, there's CWD present in urine, and there's CWD present in feces. With urine and feces and saliva, it's not very much. but after a while in the environment, particularly with feces, it really starts to add up the environmental burden. So once it gets in the environment, it's a long-lasting, persisting protein, which makes it really different than other proteins. It's extremely resistant to degradation. So we know that things like fomites, feeders, waters, fence can be a reserve for reservoir for CWD. I did a study in water to show that it does show up in water, mostly attached to the fine particulate matter like colloids in the water. And this is a scanning electroscopy image of a colloid clay particle. And you can see there's lots of little cracks and crevices. Prions love to stick to things like that. So this has a little bit of a negative charge. Prions have a little bit of a positive charge. It's a match made in heaven, and it sticks to these soil particles. And then we have plants, and I have a question mark here because in laboratory studies, a number of different labs have been able to show that plants will pick up infectious CWD prions in the roots and in the aerial tissue, so the leaves and stems. What we don't understand is for sure what's happening in the real world, because the soils and concentrations they're using in the lab are not like those in the real world, where we know there may be some sequestering of the prion material in soil matrices. So that's still a question. In the real world, is it getting into plants? If so, how much? Are monocots and dicots could be different. And is that happening in all locations, or does the soil profile matter? And I think soil profile matters from what we've looked at from other studies. So as far as indirect exposure routes, you have ingestion through eating. So here's a little spike. He's having a hay snack. So if you have a food source where animals can get at it, you could easily transmit disease between wild and farm cervids. So you should never have feed, hay, or other things accessible to wild cervids. Here we have a whitetail. He's doing a scrape, so he's going to rub his preorbital glands on the stick. Then he's going to make a little scratch down here with his hooves, and then he's going to urinate on it. And then another buck is going to come and do the exact same thing. So they definitely investigate. This is a territorial behavior during rut, a great opportunity to have multiple deer in one location. And then here is a trail cam image from Rocky Mountain National Park here in Colorado. And this is a mineral lick. And what this shows is that a lot of different species are using the mineral lick. So we have some mule deer here using the mineral lick, and we have an elk using a mineral lick. So while they're eating the soil and they intentionally consume it, we think because of salt, they're also urinating and defecating, so they're contaminating that. So here you have a way of congregating animals in one location that can spread. I also did a study on inhalation where I used dust and was able to successfully transmit disease via dust. And actually, it's a pretty good way of doing it. Out here, it's very, very dusty. And so when these animals are in rut or they're running, they stir up dust particles, or if they're snuffling in the soil, that could be another way they're picking it up. Humans doing things they might not oughta. That's the theme of this slide. So this is a mortality pit here in Colorado, where they put stuff from the fisheries. Here we have a deer carcass, and they put a trail cam out here. And there's another great study that put a trail cam on a deer carcass in a wooded area, and it'd be easier to say who doesn't come to check out the carcass. And one of them, there was a robin standing on the bloated carcass. So deer are super nosy. So here we have deer, and here's a little fawn, and they're coming to check out this dead one. Here we have lots of bird scavengers. We've got a bald eagle, we've got some crows, got some magpies. They're all coming to snack as well. So if you leave this stuff out in the open, you have the potential translocation via scavengers. So what happens when CWD gets inside of the animal? So I like to use this little diagram, and the easiest thing for you to remember is front to back to front. So here, these little spiky red blobs are the infectious material. It comes in the animal either nasally or orally, probably more likely than not, the dominant way would be orally. And then it's immediately shuttled to the regional head nodes during just normal pathogen sampling. So you've got an animal that's constantly looking to make sure there's no pathogens, the immune system's looking. So you have shuttling between these two areas into the regional head nodes. Now you might think, well, the brain is only a mere couple inches away. Surely it goes there. It doesn't go there first in deer. So there are a few things that climb up these olfactory sensory neurons. Polio does it, rabies does it, CWD does not do it. So once it gets in the head lymph nodes, that's why we collect the medial retropharyngeal lymph node. That's a extraordinarily essential sample for testing for CWD, because that is one of the very first places where we have proliferation of CWD in the animal. Once the infectious particles come in, they go in and start converting the normal endogenous prion proteins. And we don't know exactly how this happens. Like I mentioned before, we have a protein that's under normal conditions quite stable. Something causes the protein to change conformation. And that happens in these tissues like the lymphoid tissue. So in there you start having a buildup of the abnormal form. And at some point, and it's variable depending on the genotype of the animal, it starts moving along to the other parts of the body. You can find it in the blood somewhere along this path line. The timeline's variable. You can find it in the spleen. And back here is the rectal tissue. So there is a ring of lymphoid tissue on the rectum, and we can find CWD there as long as it's had time to go from the head to the rectal tissue. Once it goes there, then it makes its way up to the brain. So 1, 2, 3. So during this process, we see shedding, and there's data to show that the shedding occurs rather early in the animal. So there has been CWD found in feces, urine, and we have found it in semen. Although we don't know the consequence of that yet, our partners at USDA Agricultural Research Service is trying to investigate that for us to understand, okay, we found it there with amplification assays. What is the significance of that, and can it transmit disease either to the dam or the offspring? So stand by on that question. We're hoping to learn the answer to that with their study. But as I mentioned, this is a slow progressing disease, so it'll be some time before we find that out. And then we also see shedding in saliva. But during this, you can find it all over. When an animal is on the terminal stage of disease, you can find it most places you look. But prior to that, not so much. So that makes antemortem testing challenging. Another thing that makes testing and detection challenging is genetic variability at codon 96 in white-tailed deer. It influences the proliferation, so it influences how quickly this all happens. The glycine animals are wild type. They're the most common that occur naturally. And the CWD prions proliferate and traffic the fastest. And this is ultimately where it's coming down to, is how quickly is that proliferation and conversion to the aberrant form happening. In the intranasal study we did, we found really good examples of that between these 3 genotypes. When we counted positive follicles in lymphoid tissue, it was very different. So GG had the most positive follicles. These animals are going to get sick the quickest. That's not taking into account some of the more complex whole genome predictive genetics we're talking about later in this presentation, but just as a rule of thumb, you can keep this in mind. None of these genotypes are truly resistant to CWD. And I keep seeing things that says they are not. We have seen plenty of SS animals that have CWD, not just 1 or 2. So they absolutely can get it. The difference is how long it takes them to get to that clinical stage. SSs are very, very slow. And then GS is kind of an intermediary. When we think about trafficking, one really important thing to remember is that deer and elk are absolutely not the same. The progression is different, the duration of progression is different, and the sites of deposition can be different. And we really don't understand exactly why that is. So when we look at our graphic again for the elk, the same progression I talked about before is still true. You still can find the front to back to front, where you have you could have an animal where it's lymphoid positive tissue, positive only in the lymphoid tissue in the head in the medial retros, but the brain can be negative. That's very common in deer and elk. However, there are a handful of elk that have decided they're not going to follow the rules. So anywhere between probably 8 and 12%. I've seen various numbers looking at our data and those of other folks. You will see elk that are positive only in the obex and not positive in the medial retropharyngeal lymph nodes. And we have absolutely no idea why that is. It does not appear to be dependent on the prion, the dominant prion codon, which is 132 in elk. We really don't know why that happens, but it does. So having confidence in your CWD testing in elk, you must absolutely be looking at both tissues. Because if you don't, I have seen some folks that say, oh, well, we're only going to test, and not for our program because that's not allowed. We're only going to test the medial retropharyngeals. You might be able to get away with that in white-tailed deer. There's some reasons why you want both in white-tailed deer regarding epidemiology and so forth, and sometimes tissue matching for DNA. But with elk, if you don't do that, you're going to miss potentially 10 to 12% of your positives. So you don't want to do that. These guys also shed. We have not looked at semen in elk, but I have a strong suspicion it's probably there as well. And then we've got this new twist, and we kind of knew it was coming, and it's become more visible in the last probably 5 years or so, and that's the idea that there can be strains of CWD. We know there are strains of BSC. We know there's strains of human prion diseases, and it was suspected that they were also present in CWD, and they absolutely are. And this adds another element of complexity to the whole problem because they don't always behave the same. So there are a few ways to look at this. And one of them is actually the migration of the protein on a Western blot. So that's what you're seeing here. So in this lane, we have a standard CWD sample. And with deer and elk, they have more diglycosylation. So 2 sugars, they still have 1 sugar, and they have some that is unglycosylated. But that's one way we can look at a difference between species. So each one of these bands represents the amount of glycosylation on the protein, or glycosylation is a fancy word for sugars. The smaller the protein, the faster it runs. So the smallest proteins are down here, the segments, and then here and here. So unglycosylated, monoglycosylated, and diglycosylated. What you see, here's our standard CWD, but you start seeing some changes. You start seeing a little bit of a band shift, and some of this is rather subtle, and it can depend on the quality of your gel, but that's one way you can kind of look. Some of the shifts are quite dramatic. So this here, you can see it's clearly lower than these. So this is lighter. This is shifted down. And then you'll see sometimes a change in the dominance of the glycosylation pattern. You might see more of the diglycosylated versus the monoglycosylated. There's another way to look at this. It's for the stability of the protein. Some of the prion proteins are more stable than others. That's considered a characteristic of strains as well. So it adds a level of complexity, and we don't fully understand the consequences of it. I published a paper on elk that there were 2 different strains here in the Rocky Mountains. One seemed to be very aggressive, where it killed all the elk kind of without exception. It even went after the LL elk, which are leucine. They're kind of the slower progressors of the elk world. They have a lot of durability. It even went after them. It took longer, but it did it. The other one was much more slow moving, and it had a much lower prevalence rate. So that's kind of what we're seeing as far as maybe what it does on the landscape. It may affect incubation period and general attack rate. But now they don't always look different. I should add that sometimes you can see a different Western blot profiles, but not always. So in the elk study we did not see a difference in the Western blot profiles. So how do we find CWD? Well, the most dependable gold standard is postmortem, and it's with immunohistochemistry. So we look for regulatory samples, the OBEX, which is this area right here at this bifurcation. So if you're collecting these, it's better to include more than less in your samples. If you're throwing them in a jar of formalin, you know, you're better off to go from here to here to make sure you've got this area in here, because we see a lot of issues with collections on OBEX. Folks have a hard time getting the right region, and so then it's untestable. We also require that both the left and the right medial retropharyngeal lymph nodes are submitted for our program. We require half of those to be fresh and half fixed, so right down longitudinally of these nodes. And the reason we want both sides, and there's going to be a paper coming out on this, but I've known it for years, which is why we wrote our program that way, is in early cases you can have unilateral distribution of the prion protein in the lymph nodes. So you don't want to miss it. So if you put both slices in there, both sides, your odds of finding it are going to go up. This image is just a IHC slide. You'll see the infectious material sometimes represented with a brown antibody, sometimes it's pink. So you can see here, lots of stuff going on. There's some spongiform degeneration here and here. We also have some approved whole herd antemortem diagnostics for our program, but they're very selective. And so if you think about when I mentioned that there's a lag period between the infection in the head and finding it in the rectal tissue, that's where we run into some trouble. So we don't look at antemortem testing as an individual animal basis because you can miss it. So when we look at antemortem diagnostics, we look at whole herd. We've crunched the numbers with the sensitivity and specificity of rectal biopsy immunohistochemistry and kind of determined, you know, what is acceptable for us. And we use it in specific situations to reduce quarantine time for trace out or exposed herds. This is not an approved method for CWD positive herds. The pro of a rectal biopsy is it's easily accessible. You can evaluate it by HC. We know the sensitivity in all the genotypes, except for SS, that's a little fuzzier. You don't have to do chemical mobilization, although I do. I can't, I have seen a colleague of mine, he's so good, he doesn't, he can just do them in the chute. There's no way I could do that, because once you let go of your little pinch with your rat tooth forceps, it bleeds and you can't see where you're going. And you use disposable instruments between each animal, so you are not passing CWD along. Prions love stainless steel, so you can get transmission between animals with your instruments. This is really, really important for you that work on cervids, where you might be able to toss your instruments in Vicron. If under normal circumstances for other diseases, you cannot do that with prions. You will infect other animals that you use those instruments on. So disposable instruments only. The downside of this is that there is a level of skill required. There's a specific region that you have to take the sample from because that band of lymphoid tissue is rather small. We must have at least 6 follicles to make sure that the test is valid. The sensitivity isn't great. It goes down in those GS and SS animals. That's because they may be positive in the head nodes, but we haven't found it yet in the rectal tissue because it takes longer to get there. The rectal tissue becomes positive later in the disease course, so you're going to miss those really early cases that if you were to euthanize them, you might find it in the medial retropharyngeal lymph nodes. There are a limited number of samples you can take per animal. There's one study that suggests that lymphoid follicles do regrow. We weren't sure about that for a really long time. However, the ones that regrew after they were already detected to be positive, those new ones hadn't gotten positive yet. So there's a limited number you can do. And then as animals age, they tend to have fewer follicles. So up here in this corner, you can see there's a nice lymphoid follicle. And here we have staining. So the identification in lymphoid tissue is not just the staining, it's the location where the staining is. Like if there was staining out in here, they would probably call it artifact. It needs to be in the lymphoid follicle. So there's more than just the presence of staining, it's got to be in the right spot. So stopping the disease, this is really, really difficult, if not impossible. And so some things that make that hard is that long asymptomatic period. And during that time, they're shedding CWD and excreta into the environment where it stays. So it doesn't degrade. There was a study that was done on sheep scrapie, and it was like 15 or 18 years later, they'd removed scrapie positive animals. They figured after all that time, it'd probably be fine to reintroduce sheep. It wasn't. They put the sheep there and they got scrapie again. So we don't really know how long CWD can last in the environment. There's been some work done, um, but it's not encouraging. It just shows, yeah, it's really difficult to get rid of. It's really resistant to deactivation, both naturally, as I mentioned. So UV or microbial action is not going to do anything to it, and disinfectants don't work. The only thing that kind of does is bleach. It has to be at the right concentration. And even with instruments, I don't trust it to completely get in all the cracks and crevices. Normal decontamination procedures don't work, like autoclaving does not work. So that makes that difficult. And then we have problems finding it early in live animals because of that lag period. So what about wildlife? Different agencies have tried different approaches, targeted culling, carcass movement restrictions, baiting restrictions. So anytime you're gathering animals together, that can't be a good thing for disease control. Increase in surveillance from hunters, and not allowing dumping and trying to discourage dumping. So this is a picture someone sent me from the Midwest that they saw along a back road, and someone dumped a deer, and then his friends thought it was all a good place to dump the deer as well. So here we've got lots of spinal columns. You know, if any of these were positive, the scavengers have moved this all over now. So that's why there's a lot of carcass movement restrictions, like you can't have bone-in meat. They're limiting things that can come in and out of the state because they know if someone comes, they bring a whole deer, they're going to dump the gut pile out back. Deer have been reported to munch on gut piles. So those things you want to avoid. So with farm cervids, we have, I think, a greater chance of getting a grip on the disease. So participation in our voluntary herd certification program is a step in the right direction. We require 100% mortality testing in animals over 12 months of age, and they have to be certified in order to cross state lines. So they can't move across state lines legally without being certified in our program. We also strongly encourage producers to practice good biosecurity measures to keep it out of their premises. Another thing that we're working on is predictive genetics with CWD susceptibility. So as I mentioned in a previous slide, while the prion polymorphisms of the prion gene impact incubation period, it's clear that it doesn't prevent it. So we wanted to look a little deeper. I mean, biological organisms are complex, so we wanted to look at the whole genome. There could be factors biochemically as far as destruction of proteins or recycling or any number of things that might have an effect. And sure enough, that's what happened. We funded Dr. Chris Seabury at Texas A&M. He is a geneticist because I am not, and I don't even pretend to be one. It's a very complex field. So we really needed someone who knew what they were doing. So we gave a cooperative agreement to Dr. Seabury, and we looked at a huge number of white-tailed deer samples. We looked at over 1,000 for the first group. He used machine learning and started to look for single nucleotide polymorphisms that were different between the CWD positive and non-detect animals. So what we found is there were over 200,000 SNPs that were initially checked, and he settled on about 124,000 of those areas. And what he further found out from that there's a suite of several dozen genes that seem to have the most influence over susceptibility. So when he puts that through an algorithm, he can get scores for each animal and kind of put them in 3 different categories: highly susceptible, moderately susceptible, and minimally susceptible. So this is what we're moving towards with the farm cervids, is we want to encourage producers to test their animals. And I'm trying to see if I've got another, oh, there's a blank slide that I forgot to fill out. So we further made this more affordable. He whittled that 124,000 SNPs down to 50,000. And it made it so it was $75 a sample for producers. And they get their predictive genetics score. So they can look at all the animals in their herd and tell which ones are highly susceptible and remove them. Also, don't breed them. Try to breed for the better scores. At this point, we're at about, I think, 35,000 deer in the US have been tested, but we encourage producers to keep testing. There's the North American Deer Registry that is facilitating that with a company called Neogen. So they facilitate that so producers can get that data and we can start making a good swipe at the disease. So if we can get rid of the highly susceptible ones, that's going to help prevent the point source introductions. We don't know what'll happen if there's already a high prevalence on site. But part of what we got is, like, I can think of a herd where we had a 75% prevalence. 25% of the animals after all that time still were not CWD positive. So those animals might have made it in the long haul and they had good scores. So that's what we're kind of trying to hope to achieve to keep CWD out of the herd in the first place. Oh, and here's my slide. So encouraging selective breeding. And that is all I've got, but I'll be happy to answer questions.
Hi, this is Tina Cookingham, Animal Health Inspector, and I'm just going to guide us through any of the questions in the chat. And so far, there was actually just one that was related to Dr. Beth Ann Shane Holzer's presentation. And the question basically was, have Reservis been removed from the list of CWD susceptible species in New York?
I just posted the answer for everyone with a link. Dr. Sykes, thank you. You are right. My apologies to everyone. New York does use the USDA's definition of cervid for our CWD susceptible species list, and it doesn't include Reservis. Actually, I don't know if Dr. Nichols, if you can talk to one that was removed. Do you know?
We have not changed anything on our CFR. So in our CFR, here's why it's kind of a weird list. So when they first developed the CFR, the rule for CWD, at that time, the only species that we knew definitively to be susceptible to CWD were deer, elk, and moose. Since that time, a lot has changed. However, the language wasn't written in the CFR to make it easy for us to expand that list should science change. So we know that reindeer are susceptible. We know that muntjac are susceptible. We know that cyca are susceptible. But they're currently not required in our program. If we open the CFR, then we're going to make the language more broad so that we can make changes to the list of susceptible species more easily.
Thank you. The next question is, do you think it is safe for dogs to eat venison diets?
Oh, I guess we don't know of any cases where dogs have become infected with CWD. There's always a first time, right? But we don't know of anything like that. Where is the venison coming from? I know for human consumption, a lot of it comes from red deer from New Zealand. So I don't know where they get it. We don't slaughter a huge amount of cervids in the United States. Considering how many we have farmed, the amount that actually go to slaughter is very tiny. Most of our industry is for the hunting business versus the meat business. I guess, you know, if you're wondering about that, contact your dog food company and see where they get their venison. Because venison is generally cervid meat and materials are not rendered in the United States. Most renderers want nothing to do with it because of the CWD issue.
Great, thank you. The next question is, what percentage, if any, of the herds in New York State are currently affected with CWD?
I am not aware of
Hi,
any.
this is Yeah, I'm happy to report. This is Dr. Shane Holzer again. I'm happy to report none. We have currently have none. Our last case of CWD was back in 2005, and we had a strong response, and we have had no CWD since then.
All right for that. The next question is, what recommendations are made to hunters to prevent human exposure to CWD as they look healthy until late in disease?
Yeah, so the CDC has come out with recommendations for hunters. So if you go to the CDC page and put in chronic wasting disease, you'll find um, parameters and recommendations. Currently, they recommend that if you're hunting in a CWD endemic area, they recommend testing prior to consumption.
And the next question is, what happens to the land where a captive herd is diagnosed with CWD?
It's a wonderful question. So in most states, there's the 60-month, 5-year quarantine time that's placed on that property. But what happens after that when we know that there's this extended period where we don't have degradation of CWD? I have not seen any states able to enforce anything longer than 5 years. So I've seen states purchase land. So there was one spot in Wisconsin that was really, really highly infected. The DNR actually purchased that guy's place and they've kept the fences up. But I'm not sure states have been able to fight that very well. There's even one state that the guy wanted to take his fences down sooner than 60 months, and I think he might have won that lawsuit.
The next question is, what bleach recipe to clean a hunting knife or etc. If a hunter doesn't consider disposable?
Okay. Um, we recommend it's 1 to 20,000 parts ppm of bleach. So I think if you look up, I'm trying to think if APHIS has that. We have it in our documents all over the place. How easily they are for other people to find, I don't know. But there's a recipe, I think, floating around for that. I know I've provided it to some states. But bleach is a funny thing. Like they tell you not to use bleach if it's over 1 year old. Apparently the chlorine degrades, even though it smells bleachy. It's not as effective. So 1 to 20,000 parts per million, and it has to do with the concentration of the actual active agent kind of varies. So if you did, just if you want to do something quick and dirty, if you did a 60% bleach to a 40 water, you're probably okay.
The next question is, which state in the US is CWD most prevalent in?
Oh boy. Let me ask you a clarifying question. Do you mean in the wild or do you mean in farm service?
We'll just move on to the next question while we wait for
Okay.
that person, I guess, to chat back. What is the current thinking on risk from deer or elk urine used as attractants, and are these current restrictions on use?
Okay, so currently the USDA, we don't have any restrictions in place for urine lures. I can give you my personal opinion as a scientist, and you can do with that what you want. So when they collecting urine for the lures, many of those herds are certified. I can't tell you if all of them are or not. They put them in bio cages and they collect the urine from the does. It's then combined into a big vat. Then they take that and it's god awful smelling and they aliquot that out into, the little bottles that they sell. Considering that it's pooled and we already know it's a very, very low concentration, I think there are much more significant things to worry about than urine lures for transmission. Now, I know a lot of states have acted on that, and DNRs certainly can do that. To me, it's kind of an easy target. Um, do I think there's a huge risk with urine lures compared to other stuff? I kind of don't. And that's my opinion, not the opinion of the USDA. That's me as a scientist. Um, that there are definitely other threats that I think are higher risk because you don't put a lot of urine on your clothing or whatever anyway. So it's kind of watered down. Um, so the question was what states have the most CWD, and she said both. So that's a great question. With wild, I'll take a stab at answering that. At this point, I suspect it's probably there's probably a race going on between Colorado, Wyoming, and Wisconsin. Although Arkansas seems to want to get in on the game as well, they have a lot, but CWD started, from what we can tell, in Colorado. So our mule deer populations here and elk, even though it's a little bit less, are pretty heavily infected. And Wisconsin, it's just taken off like wildfire. So in farm cervids, again, we kind of have a race going on. I would say probably between Wisconsin and Pennsylvania. I think Pennsylvania is, because I actually compared that not that long ago, and they were neck and neck. So both of those states have high prevalence rates in farm cervids.
The next question is, what is the risk to humans consuming deer meat that comes from an unknowingly CWD positive deer?
The quick answer is we don't know. The CDC continues to monitor humans for increased presence of neurodegenerative diseases. The question we've always asked is, would we know what CWD looks like if we saw it? Would it look like spontaneous Creutzfeldt-Jakob disease? So they monitor the incidence of CJD in humans through their networks. There's been conflicting results in animal studies. So it's a little bit unknown, and with some humanized mice. It may be dependent on why these results are variable. It may depend on the strain, which we fully don't have a good grasp on that yet. So I would say it's unknown. I think the CDC recommendations are solid. If you're hunting in an area that's endemic for CWD, just go ahead and get that tested. That seems like that makes the most sense to me. You wouldn't want to eat a diseased animal anyway.
And
How
then
about
the,
risk of
sorry, I
semen?
was going to say the last question. Yep, about the risk of semen for AI.
Great question. One that we really, really want to better understand. I wish I could say that we do, but I don't. We have got the study going on at ARS to determine if the amount of infectious material in semen is sufficient to cause disease. AI, I think, has its own set of issues besides the potential for infected semen. I'm uncomfortable with AI, I will admit it. We have people going from farm to farm, and you all know, for disease control, when we think about things from FMD and these other diseases, that you don't want to take materials and instruments from herd to herd, but that is exactly what happens with AI, whether it's lab AI or regular AI. It makes me squeamish. I have told producers, if they can afford to buy their own instruments, that it would behoove them to do so, because why not take that extra precaution?
It looks like we have another one. How are positive carcasses disposed of properly to limit spread of disease?
So there are a few methods of disposal that we have found acceptable. So for depopulation events, there's incineration, although incinerators are harder to find these days than they used to be. There is alkaline digestion. Universities, some of those have alkaline digestion in their necropsy labs. Burial is acceptable. In on-site, we always have the states look with and talk to their local EPA. There are restrictions sometimes about burying our animals depending on the water table levels. And also landfill disposal is also acceptable. What we don't want folks to do and try to discourage is the practice, and it's very common, is making dead piles out in the back 40. There's just nothing really good that can come from that.
And then looks like the last question is, what is the likelihood of spontaneous CWD?
Also a great question. These are good questions. We think it probably happens. How often? We just don't know. It definitely happens in other species. And I mentioned spontaneous CJD in humans. Finding it is tricky, because what would it look like compared to normal CWD? Could we differentiate it by our normal diagnostic processes? What we've tried to do, and it's like the science gods are working against us, is with our program, we require folks to submit both fresh and fixed tissues, and that's sometimes really difficult to get people to do. But it's important because if we have an animal that shows up just out of the blue, and occasionally we get these, where all of a sudden we have a positive animal and they never get another one, or it's somewhere that we don't expect CWD to emerge, and it may be a closed herd. Those are the ones that we are definitely curious about. And it's like fate has not been on our side because we've had a few of these instances. And I check with NVSL. I'm like, don't suppose they submitted any fresh tissue? No. So anytime I get something that looks strange, I send that to ARS because Dr. Justin Greenlee is looking at strains. And we're hoping one of these times maybe we can find something different. Could the reindeer in the US be spontaneous? Could be. Can't rule anything out at this point. It does appear to look, like our normal CWD, but that's another one that owner was not particularly cooperative about submitting fresh tissue on her mortalities. I finally got some just really recently. So we're going to partner with Colorado State University. Dr. Glenn Telling there did the work with Sylvie Bernstad in Norway, where he compared the Norway CWD to the North American CWD. And I'm going to give these samples to Glenn so he can then see how they look similar or dissimilar to both of those things. So we'll see what comes of that, whether it's spontaneous or not. I don't know if we'll be able to discern that, but maybe we'll learn something from it.
And it looks like that's it for the questions in the chat. Thank you, Dr. Shane Holzer and Dr. Nichols.
You're welcome.
Yes, this is Dr. Shane again. Thank you all so much for taking part of your day to come and listen to CWD. It's a very interesting disease as we learn about it. There's so much that changes with it, and I want to thank Dr. Nichols so much for coming and talking to us.
Sure. And
Very different.
I'm here to answer CWD questions at any time. So I'm an open resource for questions you might have. You can reach me at my email address. And I think I'm sure I'm searchable on the APHIS website. But yeah, that's part of the job that I actually really, really like doing is I like talking about CWD and um, teaching people about it. So anytime you can call me.
Thank you. Have a great day, everybody. Bye-bye.
Thank you. Bye.